My favorite days, favorite parts of the academic year, which is this special lecture by our trainees in honor of Mr. Joe and Betty Ward. We have two outstanding awardees, Stacy Hussong and Katie Lewis. Before I introduce them, I want to tell you a little bit about Mr. and Mrs. Ward, who some of you may have met already. So this lecture is called the Joe Ward Jr. and Betty Ward Award for Excellence in the Study of the Biology of Aging. It was established in 2010 through the generosity and Joe and Betty, and we are very thankful for their gift in support of this this lecture. Mr. Ward passed away in 2011. He is a Texan. He was born in Austin, and he attended UT in Austin, and then got his bachelor's, master's, and a PhD in '53, and married Betty Branson in 1949, and they moved to San Antonio and taught math at Military Institute in the Texas Military Institute. He was a pretty accomplished research scientist, and I've always reminded that sure he would be enjoying our presentations very much. He worked at Lackland Air Force Base as well as at Brooks Air Force Base, and spent 4 years at the Southwest Educational Development Laboratories. His interests were very broad: computer, statistics, and science. But also he enjoyed sports very much. In 2001, the Northside Independent School District named a new elementary school in honor of Mr. Joe Ward. So the Dr. Joe Ward Elementary School, for his many years of service to the children and the community. And Betty is very dedicated to her family, their children, Brad and Kent, and to teaching the youth of the community, also as elementary school teacher. So please join me in welcoming Betty and her son, Brad, and also for thanking them for their generosity. Our first speaker is Stacy Hussong, who works in the laboratory of Dr. Galvan in areas related to aging and neuroscience and Alzheimer's disease, which obviously are issues very important in our communities nowadays. She obtained her bachelor's in chemistry and biology at the University of South Dakota, and then obtained her PhD at the University of Minnesota, and I guess she had enough of the cold weather and came to Texas. She has been very, very productive, has many publications, and has received many awards and recognitions. So many I won't mention them all, but one of them, she graduated magna cum laude. She has been in the training grant and has received a number of travel awards and has won several presentations and posters. And she has already 10 publications, and I've seen Stacy present her work a couple of times already, and I'm very impressed. So I'm sure she's going to be a very accomplished scientist and making important discoveries. So, Stacy, please go.
Okay, thank you, Dr. Musi. So first of all, I would like to take this opportunity to thank the Ward family. This award is truly an honor, and it's a great opportunity for young researchers to showcase their work. So this afternoon, I'm going to try to give an abbreviated version of my project that shows the far-reaching effects of knocking down mTOR complex 1 just in the brain, and we see changes in overall cognition and also metabolism as well. So first of all, just to give a little bit of background about this project and where it's coming from. So knocking down mTOR complex 1 throughout the body has shown to have many different effects, especially in the brain. So we see that there's changes in peripheral and also brain vasculature. There's improvements in proteostasis, improvement in cognitive function, and also in affective disorders such as improvements in anxiety and depression. So, however, these studies and other improvements have been shown as well. However, all of these studies are shown with a systemic treatment. So you're knocking down mTOR complex 1 in all cell types. So we don't know actually what is the neuronal component that is involved in this. If we only knock down mTOR complex 1 in the brain, do we also see some of these similar phenotypes? So in our model system, we're knocking down mTOR complex 1 specifically in the neurons. And we're doing this by targeting the raptor protein. So the raptor protein is one of the required components of mTOR complex 1. And this model system is very neuronal specific, so we only see this knockdown in the brain. And it's also inducible, which is important because we only want to knock down mTOR complex 1 in neurons of adult animals because it's been shown that it's actually embryonic lethal if you knock this down during development. So to kind of just characterize our system, we looked at many different measures, including mRNA expression, mTOR complex 1 signaling, and kind of the gold standard now is looking at mTOR complex 1 formation. So we developed 3 different groups. Our controls are no manipulation, so this is 100% mTOR complex 1. We also have a 35% knockdown group, which was not significantly different than the controls. However, this is very similar to what you see with rapamycin treatment, so we decided to include it in our studies. And then we also have a 60% knockdown in mTOR complex 1. And I will refer to these as the 35% knockdown, 60% knockdown. So first of all, we wanted to see whether or not these changes actually had an effect in the whole brain. Does we actually damage the brain by knocking down mTOR complex 1 by this much? And we did not see any changes there. We also wanted to look at the overall lifespan of the animal to see whether or not this was affected. And we looked both in males and also females, and there was no difference between the two. And again, there was no difference between all 3 groups. So knocking down mTOR complex 1, even by 60%, does not affect overall lifespan in these animals. So because we're knocking down mTOR complex 1 in the brain, we wanted to look what happens in the brain. Does this affect cognitive function? So we're looking at, first of all, cognitive function by the Morris-Water maze, which is a very powerful spatial learning and memory task. So during week 1, you can see there's really no difference between the 3 groups. They're all learning at the same rate and finding the platform at the same rate. However, during week 2, we actually moved the platform to a new location. So we're challenging the animals to see whether or not they can relearn a new platform location. And in this, we actually start to see differences where the 60% knockdown of mTOR complex 1 actually has a deficit. They take longer to find the platform, and they aren't learning quite as fast. They do learn towards the end, but it takes them a little bit longer. And if we look to see where they're looking for the platform, they're looking for the platform where it used to be versus where it is now. So they have a little bit of a problem with cognitive flexibility. At the end of each week, we actually do a memory test. So just to see whether or not they remember where the platform is. So we remove the platform, count how many times they cross where the platform should be. And for this, we actually see that the 35% knockdown actually has enhanced memory over that of wild type, which is actually quite remarkable because these animals are quite young. The wild types have no deficit, but the 35% knockdowns are actually doing better than that of the knockdowns are actually doing better than that of the controls. To further underline this, we did another memory task just to see if we saw the same results. And again, we saw a similar pattern where a 35% knockdown actually have enhanced memory over that of controls. Just to see possibly what could be the underlying mechanism for this, we looked at a metabolism in the brain. So this is looking at brain glucose uptake as measured by PET. And this was done by our collaborator, Eileen Lin. And again, we see this very similar pattern where the 35% knockdown animals have improved brain metabolism and glucose uptake. And here we saw actually the 60% have a decreased brain glucose uptake as compared to both of the controls and also the 35% knockdown. So overall, there looks to be an impairment in the 60% knockdowns. So because the brain glucose uptake is actually very highly regulated. However, there could be some underlying cause in peripheral glucose metabolism that could be driving this. So just to double check there was no changes there, we looked at peripheral glucose metabolism. So we looked at fasting glucose and there was no difference there. We also looked at a glucose tolerance test, again, no difference. And also the insulin tolerance test. So overall, it does not look like we have a problem with overall glucose homeostasis in these animals. So just as kind of a summary for this first part, we see that the relationship between mTOR complex 1 formation and also in memory and brain metabolism is not linear. There isn't more mTOR complex 1 is better and less is worse. It's kind of an in-between, where actually about a 35% decrease appears to be the sweet spot, I guess, as far as mTOR complex 1 signaling. If you go further ahead and have more mTOR complex formation, as shown in the TSC1/2 knockouts, you actually have cognitive deficits. Furthermore, if you go too far the other way, which has been shown in the literature, as far as an acute dose of rapamycin basically injected into the brain, you actually completely abolish LTP and cannot form new memories. So our data agrees with what's in the literature, and again, the 35% knockdown is actually very similar to what has been shown to improve memory as far as with rapamycin treatment. Because we're knocking this down in neurons, we're actually knocking down in all neurons. It's not just neurons of the brain. We're also knocking it down in the periphery as well. So we wanted to look whether or not there was any changes there. So first of all, this is looking at tail nerve conduction velocity. So we're stimulating at the base of the tip of the tail and then measuring at the base of the tail. So it's a sensory nerve conduction velocity. And so for this, we actually did not see any changes with our three groups. However, there was an age-related decrease, which is not unexpected. We also did sural nerve conduction velocity, and this is going to be the nerve in the foot. And this was actually a motor neuron, so it's going from the brain to the foot. And for this, we actually did see differences in our groups, where the 35% knockdown were actually improved as compared to their age-matched controls, and they were not significantly different than the young. So it actually looks like a 35% reduction in mTOR complex 1 is actually preserves nerve conduction velocity. We looked at one more nerve, which is the sciatic nerve, and this was also very interesting, where both groups, the 35% and also the 60% neuronal mTOR complex 1 knockdowns, actually did better than that of age-matched controls. They weren't any different than actually the young controls, but it does appear to have a trend as far as they even have better than the young. I have yet to study this a little further with a younger group of animals to see whether or not this is actually correct. And then the last thing that we did was we looked at repetitive nerve stimulation, where you repeatedly stimulate the nerve and see if there's a decrement over time. If it gets too fast, sometimes the neuromuscular junction can't keep up. So this is kind of just a measure of a neuromuscular junction health, which is kind of important down the road. And we did not see any changes here. So there's no problem with the nerve talking to the muscle. Okay, so just kind of a summary to tie all that together. We didn't see any changes in the sensory nerve conduction velocity. However, we do appear to have a change in motor nerve conduction velocity, where knockdown mTOR complex 1 actually can preserve that function. We don't know yet why or what's the underlying mechanism, but we still need to look into that. So one of the things that we always have to keep track of when we're doing behavior experiments is looking at body weight. Some mice fatter or some mice skinnier, because that can actually have a confound as far as how they're actually behaving. And you may be measuring something else rather than actually cognitive function. So we looked at overall body weight. And for this, we actually did not have any difference. But however, when we look at these mice a little closer and look at body mass composition, we actually do have changes, where both the 35% and the 60% knockdown mTOR complex 1 are significantly leaner as compared to the controls. So one of the reasons why this could be is maybe they're eating less. So we looked at food consumption. And the 35% are actually eating more than the controls, which is kind of odd. But the 60%, there's no difference there. So next thing to look at is whether or not they just have an increase in metabolism. Maybe they're burning off those calories better than that of the controls. So we looked at overall metabolism by respirometry, both oxygen consumption and CO2 production, and kind of perplexing, both of these were down. So that's kind of strange. However, when we look at this more closely and look at the respiratory quotient, which is a ratio between the two, and it indicates what type of energy source they're using. So 0.7 is they're burning pure fats, 1 is pure carbohydrates. They're going to be using a mixture of the two. So a lower respiratory quotient means that they're using proportionately more fats as an energy source as compared to the controls. So this could be one of the reasons why they're leaner, is they're burning fats more than they are carbohydrates. Okay, so as we saw before, sometimes you don't really see changes until you challenge the animals. So you really push them, and then you start to see differences come about. So we ran these mice on a treadmill till they were completely exhausted, and then measured blood glucose, blood lactate, and then how far did they run. So for the controls, they behaved very similar to what you would expect. You're exercising, you're burning glucose, your blood glucose levels are depleted. However, our 35% and our 60% neuronal mTOR complex 1 knockdowns aren't really changing the blood glucose levels. There's no difference after exercise, even though they're completely exhausted. If we look at lactate, it's obvious that they're burning glucose because they're actually increasing their lactate levels, which is a product of glycolysis. So, and they're making just as much lactate, if not even more, than that of controls. And also, they're running just as far. They can run just as far, and even the 35% run further, so farther. So they have increased exercise endurance. So to try to kind of focus on this a little bit more and try to figure out why aren't they decreasing their blood glucose levels? What are they doing to keep their levels up? So some of the possible sources of glucose are, number one, they could be breaking down glycogen in the muscle and using that as a local energy source, it's glucose, and therefore not depleting the blood. Another source is possibly in the liver. The liver will produce glucose and then shunt it into the blood to preserve blood glucose levels. So first of all, to look at the liver, one way to test hepatic glucose production is to look at a pyruvate tolerance test. So it's very similar to a glucose tolerance test. You're just injecting pyruvate, which is one of the sources of gluconeogenesis, and then you look at blood glucose levels. So here we did see that there was a significant increase in hepatic glucose production in the 35% knockdowns, however, not in the 60s. So this isn't the complete story, but it's part of the story. To underline this, we also looked at liver gluconeogenesis genes. And we saw that there was a significant increase in glucose 6-phosphatase, which is one of the rate-limiting steps of gluconeogenesis. So overall, it does look like there is an increase in hepatic glucose production, but only in the 35% knockdowns. So in getting these results with the glucose not changing after exercise was kind of odd. So I kind of did a literature search to see if anybody else had seen these similar results. And what I came across is there is a mouse model that overexpresses muscle glycogen synthase that has a very similar exercise phenotype. They don't change their blood glucose, and they have an increase in blood lactate. So therefore, I wanted to see whether or not our animals also have an increase in muscle glycogen, and this could possibly lead to our phenotype that we're seeing with exercise. So I did look at muscle glycogen, didn't have much luck, little variable. However, there is a trend that with the neuronal mTOR complex 1 knockdowns, that there is an increase in muscle glycogen. So further on, I went to look at whether or not there was an activation of glycogen synthesis, and one of the ways to do that is to look at GSK3 beta. And when it is phosphorylated, it's inactive, so when it's dephosphorylated, it's active. So the lower means that it's more active, and there is at least a trend that both the neuronal mTOR complex 1 knockdown groups have increased glycogen synthesis. But we still don't know what is causing this. We're knocking down mTOR complex 1 only in the brain, and we're seeing these changes in the muscle. So what is the link between the two? And one of the possible things is that the brain could be signaling to secrete something into the serum that's causing these changes in the muscle. So in order to test this hypothesis, we took serum from either our control mice or the 35% knockdown mice, and took that and we treated some muscle cells in a dish. And then we looked to see whether or not this makes changes in muscle glycogen. So what we saw was actually quite surprising. There was actually a decrease in cell and glycogen content with the 35% knockdown. With our in vivo results, we would have expected an increase, but there's actually a decrease. But we don't know whether or not this is actually glycogen synthesis or also glycogen degradation. So to look at that, I just again made cellular lysates and looked at GSK3 beta and also active glycogen synthesis, and there was no difference in glycogen synthesis. So this is kind of implicating that there's actually an increase in glycogen degradation. To further kind of pinpoint what this factor is in the blood, and whether or not it could be that it's something that is missing in the 35% knockdown or something that's added, we simply mix those two together. So if it's something that is missing in the 35% knockdown, adding the two together will bring it back up to the wild type levels. If it's something that's added, mixing the two together will bring it back down to the knockdown levels. And what we saw was that it looks exactly like the wild type levels. So this is implicating that there is something that is lacking in the 35% knockdown serum that is present in the wild type, so it's able to rescue it. To pinpoint it even further, is it something big, is it something small? We filtered the serum and then took the filtrate. So anything that is smaller than 3 kilodaltons would be in our filtrate. We did the same experiment where we took that filtrate and then added it to the wild type serum, or the 35% knockdown filtrate, and added it to the wild type serum and kind of switched them around to see whether or not this also could rescue the glycogen content. And what we saw was that the data looks exactly like serum alone. So the filtrate had no effect whatsoever, suggesting that whatever is causing this change is not in that filtrate. It's something that's bigger than 3 kilodaltons, and we just filtered it out. So we still have to do some more work to see what actually is in the serum that's causing this effect. So go ahead, in summary, we're knocking down mTOR complex 1 only in neurons. But there's something that is in the serum that is causing an increase in glycogen degradation in acute setting. So at time 0, you just expose the serum to cells that have never seen this before. However, what we've seen with our in vivo studies, there's something that changes after prolonged exposure in the blood that is causing the muscle to actually produce more glycogen. So there's a change, and then that glycogen can be utilized during exercise to keep up the blood glucose levels. So an overall summary, in our 35% knockdowns, again, we do see something that is a secreted factor. However, we don't know where this comes from. We want to do some future studies to see whether or not this is due to changes only in the hypothalamus. So we'll be injecting a virus into the brain to knock down mTOR complex 1 only in the hypothalamus to see if we see a lot of these other peripheral changes in the metabolism. Seen this. So overall, we have an increased glucose availability. And neurons, we do see that we have enhanced cognitive function, and also we have increased brain metabolism, and also preserved cerebral blood flow, which I didn't show, but we do. And then all of this together leads to an enhancement in exercise and endurance. However, in the 60% knockdown, so if we knock down mTOR complex 1 too much, we don't actually, ooh, what happened here? Okay, oh, something, there we go, skipped, there we go, that's okay. In the 60% knockdown, so if we knock down mTOR complex 1 too much, the neurons aren't as happy. So we actually have impaired cognitive function. We still have all of these changes in the peripheral glucose metabolism, but overall that neuronal component does have an effect, and these mice can't run as far as the 35% knockdowns. So with that, I'd just like to conclude and thank everybody in the Galvin lab, particularly Veronica, and also future lab members, our past lab members, Raquel and Jonathan. We've really been working hard on this project, and hopefully we'll be able to get it done and out the door. Also our collaborators, and of course I would like to thank our funding. And I'll take any questions. Yes?
How many times have you done the exercise?
Just twice. So, yeah.
The effect is very consistent.
Yeah, it's very similar. Yeah. Yes?
The 35% and 60% refer to the raptor?
Yeah. It's actually, those actual particular numbers come from mTOR complex 1 formation. If you just look at the raptor mRNA expression, it's about a 20% reduction and about a 40% reduction. No, it's actually the actual complex formation. So we IP for mTOR and then look at the ratio between raptor and mTOR. 35% less raptor. Yes, yes, yeah, the ratio between raptor and mTOR, yeah.
What happens to TOR2?
Good question. I've been thinking about this, and I haven't looked, actually. It's something on my to-do list, but yeah, you're right. There may be a change, but if anything, it's going to be up. It would be my guess, but we actually have not looked at that yet. Definitely on the to-do list, though. Yuji.
Well, I think this is a very exciting observation, but what could be the potential of the clinical relevance, the potential of the significance for the human health?
Right. Obviously, yeah, we can't make a genetic human. You're right. However, it can help target where we want to knock down mTOR. If we want to target more of the brain. It also has implications as far as how low can you go. If you knock down mTOR complex 1 too far, you're actually going to start to see deficits as far as neuronal function.
Do you think that your discovery can be potentially helpful for some of the motor neuron disease for the preventing the motor neuron disease?
Actually, yeah, I never even thought of that. We actually just kind of discovered, looked at our data more closely yesterday. It's like, oh yeah, it's only the motor neurons we're actually seeing this benefit and not really the sensory neurons. So yeah, it very well could be that if you're, targeting, or it could be even, I don't know that, has anybody looked at nerve conduction velocity with rapamycin? I'm actually not sure. But it very well could be, yeah, that it could help with nerve diseases as well. Yeah. Okay, Nat?
So your factor was, it didn't go through the filter, right? So
No, yeah, it was something bigger that it didn't go through the filter. We have no idea if it's a protein, a lipid, whatever. Yeah, that's something to do in the future. Yeah, Dr. Fisher.
I was thinking about it, the factor could be kind of like increased glycogen breakdown, increased QPCR, and then fat degradation or fat beta oxidation. It kind of makes me think about glucagon.
Yeah.
And I'm wondering if there could be either changes in glucagon levels or changes in PKA activity.
Right. Yeah.
One of the prime downstream factors.
Yeah, that's definitely something we can look at. Yeah. Brandy?
So you mentioned earlier the thought that 35% was about what we saw with rapamycin.
Right. Right. In the brain, at least, yes.
Very lucky.
We were very, very lucky, yes.
We were very lucky.
Yeah.
To see anything.
Yeah.
Because people might have
Gone too far or something, yeah.
But the other question, I had a question about the cause for the differences in body weight. Did you look at locomotor activity?
We did look at actually just basal activity in the activity cages. It was actually reduced. It looked very similar to that of the metabolic activity. We also looked at grip strength, rotor rod, and there were no differences in either of those two. But yeah. Adam?
Following up on Randy's, the 35% knockdown with rapamycin is with classical 14 parts per million
Yes.
dose.
Yeah.
Have you looked at the brain with a higher dose? It seems to extend lifespan even longer, or even
We have
borderline
not.
studies
I
now
don't.
look like it's huge levels.
Does anybody have those brains?
Do you have those proteins in the brain?
And just, with that,
Yeah. Whether or not, if it's going a little further. Yeah. Yes.
Are there any differences in the mouse model from using it on the human?
Yes, I actually, I didn't bring that up, but all the data that I showed was actually males. We also tested females for a lot of these. We saw no phenotype in the females. It was, it's very interesting, that you bring that up. But for some reason, the females did not show a phenotype. Okay, thank you.
Hey, Nick,
Hey,
can you go upstairs down on my campus?
Yeah, Shane.
We have no signal out here. I ran the control room, and maybe they can hear me via this mechanism. But can you guys get the signal back on out here, please? And for people down on main campus, just remember to use the button so we can hear the questions out here, please. Thanks. Nice job, by the way.
Thank you, Shane. Yes, Dr. Hussong was a fantastic, fantastic presentation. Thanks so much. And we're going to follow up with another outstanding trainee, well, not a trainee anymore, Dr. Kay Lewis. She obtained her bachelor's in biology in Penn State. And then she came to San Antonio, also running away from the snow, and trained for her PhD and received her degree under the mentorship of Dr. Boffenstein. And after getting her degree, She worked with Dr. Galfond and Dr. Buffenstein doing a postdoctoral fellowship, and more recently, she joined Calico with Dr. Buffenstein, which is a leading private institution in aging research. So I'm sure she'll continue doing outstanding work in California. Looking at her, a number of honors and awards is also very impressive. Just in about 10 years, she has about more than 20, so about 2 awards per year. That's very, very impressive. About 15 publications, papers, and 1 book chapter. So, and she just got her PhD, so she's doing it extremely well. I've also seen her present a couple of times, and she is very mature, very well trained, doing outstanding work. So we're very proud of you as well. Want to use this one?
No, he doesn't.
No.
I won't migrate too much. Okay. Yeah. Oh, yeah, I am kind of short, huh? What'd you say? Okay. Thank you, Dr. Musi, for the excellent introduction. And first of all, thank you to the Ward family for this fantastic award. When I started here in grad school, the award still hadn't existed. So probably about my second year, we had our first presentation ceremony. So, you know, knowing kind of the fantastic students and postdocs that have gotten this award previously, it was really something to work towards as I was going throughout the program. So it's really a huge honor for me to be able to receive this award today. That being said, I'd like to present some of my dissertation work where I studied stress resistance as well as longevity and cancer resistance in these unique little animals, the naked mole rats, which are the longest lived rodent. Okay. So these animals are naturally found in the eastern horn of Africa. They're about the size of a mouse with an average body weight of 30 to 40 grams. They're kind of like a chubby mouse, I guess. They've been a little supersized since they came to Texas, so they were a little bigger than they had been previously in other locations. They're also eusocial, meaning that their social structure is more similar to that of a termite or a bee, where you have one breeding female or queen, several breeding males, and then the rest of the animals in the colony are non-breeders, you know, almost non-distinct in a way that you can't even tell the males and females apart. So very sexually monomorphic, as it's called. They're also strictly herbivorous. In the wild, they like to go underground and chew on tubers like potatoes, or sweet potatoes are their favorite, that grow underground. And that's what we feed them in captivity as well, in addition to some other fresh fruits and vegetables. So as I mentioned, they are the longest lived rodent, and throughout this very long lifespan, they also experience a very prolonged health span, meaning they're very healthy late in age. In fact, you can see here, so here are some pictures, and these were actually taken about 2 years old, so you can even add 2 years. All these animals are still alive, so you can add 2 years onto these ages. And these are all breeding females, and you can see just by looking at them, you know, other than they've gotten a little lighter, maybe a little bit more wrinkly, they look pretty similar. In fact, if I saw these animals in a cage and I tried to decipher, you know, who was oldest versus who was youngest without knowing the exact information of the animal, I really wouldn't be able to tell because they age so well. And as I mentioned, these are breeding females, meaning that they can even breed very late into life. So this was a 28-year-old when I took a picture of her. Now she's 30 and she's still producing litters every 3 to 4 months. I don't know if that's a good thing or a bad thing, but they can do it. It's definitely an interesting fact. But also during this very long lifespan, or for most of it, they maintain activity. They're able to run around the colony. They have great body composition, so you don't see sarcopenia or the loss of muscle mass until very late in life. They maintain cardiac function as well as their metabolic rate, and pretty constant hormone levels throughout their life, as well as molecular function, so maintenance of their genome or their proteasome. So, specifically, as I mentioned, we don't observe any neurodegeneration, we don't observe a menopause, and interestingly, we also have never really observed any type of spontaneously occurring cancer in these animals, or really any induced cancer, as we'll learn later. So they're very naturally cancer resistant. Over the past 30 years, with the thousands of animals that my mentor, Dr. Buffenstein, has studied, there's only been one incidence of leukemia, which happened within the past few years, so it's pretty impressive. Now, all of this is very surprising because they have very high levels of oxidative damage. So kind of just to review, oxidative damage and oxidative stress. So reactive oxygen species, or free radicals, are a natural product of a cell, whether the mitochondria are generating them. And these can eventually lead to the accumulation of oxidative damage. And what's interesting about these animals is that they actually have a lot of oxidative damage from a very young age. So we see increased damage to their lipids, their proteins, and their DNA compared to mice, which was very surprising because this increase in damage has been shown to lead to impaired function and potentially a rapid aging phenotype. So maybe you live not as long when you have more damage. However, these animals have the damage, but they're living quite a long lifespan. And so some of the first studies, so before I had joined the lab, they had looked at antioxidants, which can scavenge a lot of these free radicals and prevent the damage from forming. And maybe not surprisingly in the naked mole rat, they really didn't have impressive levels of these things. Things like catalase or SOD1 or SOD2, they were about even with a mouse or a little bit lower, in fact. So we wondered, how are they able to live such a long life and have such a healthy life, but really, start out with this high level of oxidative damage. And as I mentioned, they're resistant to cancer. Even with this high damage, they're still also resistant to a number of toxic compounds. This is a kind of studies that I've done over the course of my PhD, where I took cells of a mouse and cells of a naked mole rat, and I cultured them, and I treated them with lots of different toxins. So this first group, you're seeing a lot of resistance in naked mole rat cells compared to the mice. The LD50 is the dose at which 50% of the cells will die. And you can see when I'm looking at all these compounds, including heavy metals or DNA damaging agents or chemotherapeutic compounds, you see a resistance anywhere that's about 2 to 15-fold higher in naked mole rats compared to mice. So they're incredibly resistant to a pretty wide array of compounds. However, they aren't resistant to everything. It's interesting because from the last slide that we looked at, that when you treat with compounds that really generate a lot of free radicals, they're really not resistant to it. In fact, they're a little more susceptible. Also, compounds that cause stress to the endoplasmic reticulum, they're a little more susceptible to that. And what's also interesting, when you expose the cells to UV light, just the cells, they're also very susceptible to that. The naked mole rats are. So continuing with that, so that was in vitro, done in cells, but I also studied some toxin resistance in vivo. The first toxin was acetaminophen, and acetaminophen is the active ingredient in Tylenol. In a very large dose, it's very toxic. It's the #1 suicide drug in the US for a reason, because it's very potent. And when I treated naked mole rats and mice with different doses, what I found was that the naked mole rats were more resistant to mice. So it took a dose of about 1,200 milligrams per kilogram to be lethal in the naked mole rats 24 hours after injection, compared to the mice, which was 800 milligrams per kilogram. And when we picked a sublethal dose, then to kind of look at this toxicity response, so a dose of 400 milligrams per kilogram, where both of them were surviving for several days, we saw that there was signs of increased damage in the mice versus the naked mole rat. So we measured 2 liver enzymes that have been indicative of liver damage, so ALT and bile acids, and both of these can be released into the bloodstream when you have damage to your liver. And we see increases in mice within 24 hours after the acetaminophen exposure, but we don't see that increase in naked mole rats. And we've done additional work to this. We've looked at the liver histology, and it looks like there's less damage in the naked mole rats compared to the mice. So they are very resistant to acetaminophen. I mentioned that they were cancer resistant. I've also tried to induce cancer several times. This is using a skin carcinogenesis method. So we put a compound called DMBA, and this carcinogen initiates mutations, DNA damage. And this compound, it's when you hear that you're not supposed to eat barbecued foods, or burnt marshmallows, which are my favorite, because they might cause cancer. This is what they're talking about. So you treat once with this DMBA, and then you treat twice weekly with TPA, which causes inflammation and hyperplasia of the cells, leading to tumor growth. So here are mice and naked mole rats. These are the controls treated with the vehicle. And then after the DMBA and TPA treatment, you can see that all the mice, and this was by about 14 weeks, had tumors. So you can see lots of little papillomas. They had some abnormal hair growths and some of these little dysplastic nevi. However, in the naked mole rats, we saw no tumor growth, no hyperplasia whatsoever. In fact, we actually saw the opposite. Looking at mice, so this purple layer here is the epidermal layer, which in mice skin treated with DMBA and the TPA regimen, you saw significant thickening. And this is what's typically been seen in the literature. However, the naked mole rats, why they start out with a very thick epidermal layer, and this has been previously described, we actually see a thinning of the epidermal layer and an increase in this pink layer, which we think is dead skin. So we think they might accelerate their cell turnover to avoid accumulation of damage and potentially tumor growth. Now, I also mentioned that the UV cells in vitro were very susceptible to UV exposure. And we wanted to see, well, and also another question when we're treating with a lot of these chemical compounds, well, can the mole rats even metabolize it? You know, are they even metabolizing it into something damaging that's going to cause cancer or cause a toxic effect? So when you use UV, as we all know, when we go out in the sun, we get sunburned. There is no metabolism of UV. It causes the damage, and that's it. So we wanted to see what happens in the whole animal. Will they still be tumor resistant? And so here are our control animals. And then when we exposed the animals to UV, all of the mice had tumors. So you can see some tumors here and here and just some dry, flaky skin. However, the naked mole rats just had a little bit of dry skin, but no tumors. And this was after 6 months of constant exposure, which was very surprising given what we had observed already in vitro. Now, because they're so resistant to a lot of things, and we know it's probably not their antioxidant capacity, we wanted to look at a potential pathway that they might be regulating better, upregulating compared to mice that might lead to a lot of this protection resistance and potentially longevity. And this pathway was the NRF2 KEAP1 signaling pathway. And NRF2 is a transcription factor, which basically means that it'll bind to DNA and upregulate the production of all of these antioxidant and detoxicant molecules to repair the cell and clear out damage. So under constitutive or non-stressful conditions, so just in all of our cells in the body right now, probably, NRF2 is typically bound to KEAP1, which sends NRF2 to the proteasome or ubiquitinates NRF2 and sends it to the proteasome for degradation. There is a small amount of NRF2 that is not bound to KEAP1 that can bind to the antioxidant response element and upregulate the transcription, and it's probably about 600 molecules at this point that it can upregulate for the repair in the cell to offer the cell or the animal some type of basal cytoprotection. So maybe it's ready to go if there is a stressor. Now, under a stressful event, so this could be an exogenous stressor, like any of the toxins that I had added to the cells, or an endogenous stressor, like a free radical, and this interaction between KEAP1 and NRF2 is inhibited. Large amounts of free NRF2 are able to move into the nucleus, bind to this DNA element, and you greatly upregulate the production of all of these detoxification molecules, offering enhanced protection. So the first thing I did was I looked at the signaling in just mice and naked mole rats. So this is actually measuring the NRF2 binding to the antioxidant response element. So if you don't have this, you're not really going to have anything up or anything down probably. And what we saw was that it was significantly upregulated in naked mole rats compared to mice. So the next step, we wanted to see, well, do they also have more NRF2? Is that why they have more NRF2 ARE binding activity? And we found that yes, they do have significantly higher levels of the NRF2 protein compared to mice, but they also have significantly lower levels of the KEAP1 protein compared to mice. So not only do they have a lot of this NRF2, it's really not being negatively regulated. So that's probably why we also see the higher NRF2 ARE binding activity. And we also measured, I also did QPCR, and we saw that same pattern in the mRNA levels at 2. So this could also be from a transcriptional level. And then just to confirm that this was really what we were seeing, this is QPCR performed on 3 genes that are typically largely regulated by NRF2. All of these were significantly higher in naked mole rats compared to mice, and some, quite a bit higher, 60 to 80-fold higher in naked mole rats compared to mice. So these are the genes that are, these are the molecules that are actually going to repair in the cell. And then we confirm that further with activity assays of each of these molecules. So GST and NQ1 activities were also significantly higher in naked mole rats. So my next question was, well, is this just a relationship between naked mole rats and mice, or is this something that's a pattern in other long-lived species? So I looked at 10 different rodent species with varying lifespans, anywhere from 4 years in the mice or hamsters, all the way up to 31 years in the naked mole rat. And what I found was, again, when I measured this NRF2 ARE binding activity, I saw a significant and positive relationship between maximum species lifespan and activity. So the longer lived you were, the more NRF2 ARE binding activity you had. And of course, everything in every animal can be attributed to body size. So the bigger you are, the bigger your brain is, maybe the higher some molecular pathways are. Everything can be attributed to body size. So I wanted to make sure that, this NRF2 ARE binding activity wasn't higher in a bigger animal, and we saw no significant relationship between body weight and NRF2 signaling activity. So this really could be attributed to the longevity of the animal. Now, I repeated what I had done with just naked mole rats and mice, and I looked at total NRF2 protein levels. However, this time I didn't find a significant relationship between maximum species lifespan and NRF2 protein levels, which I thought was interesting. So it really wasn't the NRF2 itself. However, if we remember, KEAP1 is a negative regulator of NRF2, but then there are also some other molecules that come into play. So there are a large number of proteins that can actually promote NRF2 activity other than KEAP1. And there are also some other molecules that can degrade NRF2. So I measured all of these in the 10 different species. And a few things came out. The first was P62, or sequestosome 1, which is an autophagy substrate protein. And I actually saw a negative correlation with lifespan. So the longer lived the animals were, the lower the P62 levels. This didn't really make sense in the case of NRF2, because higher levels of P62 lead to higher NRF2 activity. So it really wasn't explaining the relationship we were seeing. But it's also interesting in the fact that autophagy has been shown to be heavily linked with longevity. So there could be a story there. However, 2 proteins, so KEAP1, which we had talked about before, as well as beta-TRCP, were negatively correlated with maximum species lifespan. So the longer lived the species, the lower each of these levels was. And I further so this is showing that in both KEAP1 and beta-TRCP act to degrade NRF2 via the proteasome. And then I confirmed that it was actually bound to the NRF2 by performing immunoprecipitation. So my image is a little wonky up on here, but in mice and naked mole rats, when you looked at the total protein, so here you can see it's higher in mice compared to naked mole rats. However, when you immunoprecipitate NRF2 and see what's bound to NRF2, you can see both are present, but in fact, it looks like beta-TRCP, there's a huge amount bound to NRF2. So maybe beta-TRCP is actually a fantastic target for pro-longevity or anti-cancer compounds rather than KEAP1 or NRF2 itself. So to come back to this, this original figure looking at oxidative stress, so we think NRF2 might play a role down here, kind of after you're having a lot of this damage. It's promoting repair or clearing of these damaged molecules. So in naked mole rats, rather than have this accumulation of damage and this accelerated aging, you actually have repair of many of these things, which may lead to slow aging, cancer resistance, a longer health span, and a longer lifespan as well. So to review what I said today, naked mole rats are much more resistant to stressors, both in vitro and in vivo, to quite a few different compounds. They have high constitutive levels of this NRF2 protective signaling activity, and this signaling activity is also positively correlated with maximum rodent lifespan. So the longer lived the rodent, the higher this NRF2 activity is. So maybe this is really contributing to lifespan and a potential target for a lifespan extending compound. So to wrap it up, I of course want to thank my mentor, Rochelle Buffenstein, and her fantastic little animals, as well as the rest of the Buffenstein lab who helped me along the way, as well as a number of collaborators and my committee members, without which I definitely would not be standing here today, and then my funding as well, which was mostly from the NIA training grant for the majority of my time here. So I'll take any questions.
Yeah. ELISAs on NRF2 binding. What tissue was that, and were there with these nuclear extracts? Or,
So these were with nuclear extracts from liver.
Liver.
Yeah, yeah. I've done it in a few other tissues, and you see that for the you see higher NRF2 ARE binding activity for most tissues. Yeah. Yeah.
That beta TR, whatever, is there any any inhibitors for that right now, or is that something that's kind of like a new target?
That's so in the instance of NRF2, it's a new relationship, shall we say, with that particular pathway. But beta TRCP itself it's an E3 ligase. So, and it's been well known and well researched, plays a huge role in NF-kappa B signaling. So there might be some targets that exist within that context, but nobody's really targeted it in the context of looking at NRF2 or cytoprotective signaling.
So NRF2 levels are elevated, protein is elevated, as well as binding. How do you interpret? Is it both?
So you mean, is it regulated from a protein level, or?
So NRF2 protein level is higher than Glycomorat. And when you do the binding,
it's also higher.
It's also higher.
Yeah.
So do you think the higher binding is a function of the higher protein, or there's actually more?
So actually, and this comes from data where I've looked at other tissues as well. So not all naked molar, so most naked molar tissues have higher levels of the NRF2 ARE binding compared to mice. However, not all tissues have higher levels of the NRF2 itself. But when these tissues don't have high levels of NRF2, they basically have no KEAP1 whatsoever. So I think it's really a function of the protein regulators, and I learned this because I also tried to knock down the NRF2 mRNA expression quite a few times, and I was just seeing no effect whatsoever, and I think a lot of that is because a lot of the regulation is at the protein level. Yeah. That's an excellent question. Most commonly in the colony, they die of fighting because there are a lot of social interactions that happen. So most of our deaths are probably from other females fighting for the position to breed. And occasionally a male is thrown in there too. But the old animals, it looks like, their organs give out. They end reserve. Is it end reserve organ failure? I think Shelley has termed it. But we really don't know. Yeah. Yeah.
So that was really nice. I was wondering, since you're going to Calico, of those proteins that you're looking at,
Yeah.
which one do you think are the best targets for?
I would think the best, so I think KEAP1 is a great target, and I think beta-TRCP should also be a target of interest. I know you guys have Protandim in the NIA interventions study, right, which is an NRF2 upregulator. I remember I talked to Rich Miller about it, I think last year at GSA, and I asked him, well, what is it targeting? How's it upregulating NRF2? And he said, they didn't know. But I would suspect it's a tricky thing, and there could be other players that play a role, because if you look at KEAP1 knockdown animals, they die shortly after birth, because NRF2 also plays a role in cell proliferation. So they actually starve to death because they grow so many cells in their esophagus that they can't eat. So I think it's a very intimate system that could potentially be druggable, and there could be other players. I would like to put my money on beta-TRCP because I thought the findings were interesting, but I'm biased.
Okay, well, thank you very much. I think that was, both presentations were just phenomenal, and we're very proud of both of you, and wish you the best of luck. Kent, we have some, we like.
Sure.
Yeah, please.
I'm gonna get rid of this so I don't drop it.
Okay. So we'll begin with Dr. Hussong. Maybe.
You go for no instruction.
Congratulations. That's great. Thank you. The one will come later.
Okay, can I? Couple. Thank you.
Thank you.
Congratulations.
Yes. Dr. Lewis.
Congratulations. Great.
Fantastic presentation.